Hi, biology. This is Miss Satterley. I'm going to go over the information that you need to complete the assignment that was posted today. Hopefully, you don't have a lot of problems with it. Of course, if you have any questions, please let me know. So, I'm going to start off with what is non-Mendelian genetics? This is going to be genetics that doesn't follow the normal this or that pattern. It doesn't follow the regular dominant and recessive pattern. And there's a couple different things that falls under this category of non-Mendelian genetics. And when I'm done with this, I will post this with the assignment as well, so you have this document as well as the video. Make sure you watch the video because there are some useful information that I will say in here. So first off, what incomplete dominance is, this is when there isn't a dominant trait that is able to completely mask the other trait, like in normal genetics. So you end up with a blending of traits. So a blending of traits. So that's kind of like in art, if you have paints that you're mixing together and you get different colors, that is how incomplete dominance works. So when you have an incomplete dominance, you're actually going to end up with 3 different phenotypes instead of the traditional just dominant or recessive phenotypes. Because of this, your genotypic and your phenotypic ratios are going to be identical. And I'm going to do an example so you can see what I mean by that. And for this example, I'm going to use a flower example, the snapdragon. We're going to keep the same idea with regular Mendelian genetics, where we're going to use just one letter for this. When we talk about codominance, you'll see there's a little bit of a difference. So our flower example, we're going to talk about snapdragons. So snapdragons are flowers that they can be red and white, but they also can be pink. Red is the dominant color for snapdragons, so we are going to use the letter R. So our 3 different genotypes that we can have are going to be big R, big R, big R, little r, little r, little r. So our homodominant, hetero, and homorecessive. But our red is going to be big R, big R. When you mix red and white paint, you're going to see that you're going to get pink. So it's the same difference in incomplete dominance. If you're mixing red and white flowers, you're actually going to end up with white or pink flowers as your intermediate trait. So because of this, you're just going to have one genotypic and phenotypic ratio. We're going to do an example here. So I'm going to go ahead and cross 2 pink flowers. And I'm going to see what my genotypic and phenotypic ratios are going to be with this. So pink, if I look right here, pink is going to be big R, big R. So this is going to be my cross. So I'm going to put my parents in here just like I would with Punnett squares and fill in. So those are both big. That's hetero, hetero, and homorecessive. So I can see here that I have my cross. And now I'm going to go ahead and fill in my ratio. So yeah, my ratio is always going to be 3, which we've done. And this first one is going to be 2 big R's. And then that's going to be my heterozygous, and this will be my homozygous. So there's that setup right there. Oh, let me fix that. Okay, so I look at my Punnett square here. How many are big R, big R? I can see it's 1 out of 4. How many are hetero? There's 2. And then recessive is 1. Let's tab that over. Okay, so there's that. Now, this is the same ratio if I was going to do red, pink, and white flowers. And that's because there's whatever your genotype is, there's only one possible phenotype with it. So instead of having, if this was a normal Mendelian, your homodominant and your hetero homozygous would both be the same color. That's not the case for incomplete dominance. So that is how you do incomplete dominance, where one doesn't really cover the other. So just some other examples with this. On the homework, you're gonna there's an application question where you have to tell me. I just want to talk about some examples that you could see for this. So one of the things, if you have a red flower and a yellow flower, and when your intermediate is an orange flower, that's an example of incomplete dominance. So again, also, if you have a white flower and a black flower or fur color for an animal, and you end up with something that's gray, I'm going to change that to fur color, then that is also incomplete dominance because you have an intermediate form. Or if something is tall and short, and then you end up with a medium height in the middle, that means it is an incomplete. They're mixing together to have an intermediate range. So that is incomplete dominance. Now, codominance is different from that. Codominance is when you have two dominant traits. So a co-op is two schools. Co-captains, you have two captains. So on a team, that is what co means. It means two. So you have two dominant traits. Because you have two dominant traits, we're going to use two dominant letters. So we're going to have two capital letters. Or two capital letters. This is because in codominance, both traits are expressed, which means they're both going to be visible. And I'll talk about an example so you understand that. The same with incomplete dominance. We are still going to have a genotypic and a phenotypic ratio that are identical, that are the same. My example is horses. And okay, my example here, we have horse, where we're gonna cross a red horse and a white horse, and both of these are going to be dominant. So for my red horse, it's an R, so I'm gonna have two R's, but my white horse is a W. So I'll have two W's. And then in the middle, my intermediate is going to have one of each color. Now in this example, big R, big W, this is what we call a roan-colored horse. That means they have both red and white hairs, and it kind of ends up with a blending or a lighter pastel color looking horse when you have a roan-colored horse. In horses, you can have, this would be a red roan, you can have blue roan, which is black and white hairs that are showing up. So that is an example of codominance, where you have two distinct colors that one is not masked over the other. They are both showing up in your example here. So if I do an example like I did below, I'm going to cross a red horse and a white horse. So using my pattern, red would be two big R's and white would be two big W's. So if I put in my Punnett square here, we'll have W and W. All of these are going to be RW for each one. And the smiley homework, I kind of introduced this without talking about it. So you have seen how two different letters can show up, and you have that was kind of a little different because the two W's were actually showing a different trait or a blending of traits instead of both showing up. But it's the same understanding where we're having two letters here. So if I'm going to do my phenotypic ratio, I'm going to go ahead and type it in. 1, 2, 3, 4. And this first one would be two big R's. The second one would be RW, and last one would be two W's. As you can see from my Punnett square here, there's none of those. We do have 4 and then 0. And again, our phenotypic and genotypic ratios are the same. So if I was going to apply my phenotypes, I would have red. I would then have a roan colored and then finally a white colored horse, where red and white are both dominant. So that is our first two things. And I have one last thing that I need to talk about, and that is multiple alleles. So multiple alleles, when you have more than two phenotypes, more than two alleles. So you have to have multiple alleles where you use superscripts. So this is going to be your base allele with a superscript. I'll explain that. This is similar to how sex-linked traits work. You're going to use the letter of the trait, whatever you're talking about, and then the superscripts are going to be the versions of the traits, or what that trait would look like. And I have an example of this on the homework, and I'll go over that. There's an example in humans that we have multiple alleles as our inheritance, and that is blood types. So for blood types, our blood types are actually codominants, which means we have two dominant blood types. And sometimes they can both show up in an individual. These two dominant blood types are A and B type blood. So they're both dominant. For the letter we're going to use the letter I, and this is for something called immunoglobulin. And immunoglobulin is a type of protein that's found on the surface of red blood cells, and they come in two varieties, either A or B, and sometimes they're absent entirely, and that's when you have your recessive trait. So I'm going to go down. Our four types of blood that we have for humans are AB, A, and B. I already have this in here with the superscripts because superscripts are hard to add. So this is how the outline of what these would look like. So AB is going to be our codominant one. So I'm going to have one for A and one for B. So that is kind of self-explanatory. So that is the codominant trait. They're both showing up. Now A and B are both going to just be a single dominant trait. We fix that so it's the same. So A is going to have A. So we can have a homodominant like this. And this one right here, this is going to be, actually I'm going to take that away because now we have our heterozygous for that one. Now, B is also dominant. Instead of A, though, we're going to use the letter B. Set that up. And one of them is going to be, again, our heterozygous. And what do we have left for O? Well, O has to be two little I's. I'm going to use the same font. There's my two little I's, which means O blood type is actually the recessive blood type. So I'm going to end with an example of how you would fill in a Punnett square for blood types. I'm going to cross. This is actually, I'm going to use my parents. So my dad had type AB blood, and my mom has type O blood. So I'm going to put on the top, I'm going to just copy and paste. So he has AB, so I need to have an A and a B. And my mom is recessive. So I, little I for these. I'll use the same. And I'm having trouble copying. Copy, paste, and paste. Now I'm going to fill in my Punnett square. So this first one, I would bring my IA down in A. Well, that is heterozygous. So I'm just going to go ahead and copy this up here, copy and paste. And then that would be B blood type. And that would be the same. And I should have. So if you look at this, the offspring would either have type A or type B blood. So undo. Type A or type B blood, which means it would not match either parent. So that's my example for blood types. Now, before I am done with this, I'm going to take a moment and talk about the homework assignment. So on Google Classroom, under things to do, we have non-Mendelian traits homework. So this is the last assignment. It's due Friday at 11:59 p.m. So I'll grade this weekend. Your report cards are going out on Tuesday. So each part of this assignment is 25 points. There will be information that I'll post, which I'm posting with this now. So there's two parts. You have a Google form and then a slides, and I will show you each one of these. So if I click on the Google Forms here, and I'll just go to this, there is a matching section where I have different definitions, and that stuff is either in the notes we had previously with sex-linked traits or what I just talked about today. And then there are some examples. So here is the roan-colored horse and the snapdragons. And then this one is a problem where you'd have to figure out the percentages. I want to talk about this part right here. So incomplete versus codominance. You're going to be given an example, and you need to tell me if it's either incomplete or codominance. This first one, you have red, white, and roan-colored horses. Well, I know that roan-colored is when you have both red and white fur, which means that has to be codominance. But if we have black, white, and gray, then I know, okay, black and white are mixing to make gray, so that must be incomplete dominance since they're blending together. One thing I do want to point out is this one right here. It'll say blue, white, and blue tipped white. So it looks a little different on your end. That means that that feather is going to have white. It's white and blue. It's not a light blue. So that means it must be codominant. So you need to think about, are they both showing up or are they mixing together? The next one is on sex-linked traits, which we had the other day. So you need to answer those questions using that information. And then there's a section on blood types. And on these questions, what you need to do is you're going to read the question and do your little Punnett square. And you don't have to show me the Punnett square. You can just do it on your own to answer the question. But you'll do it the same way I did in the example where you use the IA, and so forth. Again, if you have questions, please let me know. There's a couple examples with this. This one right here, which of the following is controlled by multiple alleles in humans? Well, most likely this is probably blood types, but that is in the notes as well. So that's this portion of it. And then I want to talk about this assignment right here. Non-Mendelian traits homework. And I'm just going to present it just to show it to you. Okay, so smiley faces codominance. There are 3 types of questions. I have a codominance one and 2 examples of multiple alleles. So my genotypes are SS, CC, and CS, and star-eyed, circle-eyed, and a star-eyed circle. Now on this one, I went ahead, you were going to have to fill this in, and I filled it in for you so you had it because I wanted to make sure you had the right genotypes whenever you did the questions. So we have SS, CC, and CS here. So that's your 3 types. You're going to go, here's an example, show a cross between a circle-eyed and a circle-eyed. So I have 2 circle eyes here. Fill in my Punnett square, which the font was messed up, but it's still the same thing. And then I'm going to fill in my phenotype right here. Star-eyed, well, star-eyed has to be, if you look here, 2 S's. That's not there, so that's 0. Circle-eyed is, just to make sure, 2 C's. and these are all 2C's, so that's 100%. And then there's no star-eyed or circle star-eyed. So you have 3 problems you need to do. You're going to have star-eyed and circle-eyed, a circle star-eyed and circle-eyed, and 2 circle star-eyed. I am not grading this part. If you want to just do this on paper, you can, or you can use the Punnett square that's provided. I will be grading the percentages for it. Now there's 2 last examples here. I have multiple alleles, and I have a little description here on smiley faces, and here they are. These are the 2 types. The reason there's 2 for this is because just like blood types, you have your homodominant and your heterozygous. Based on the questions, you need to make sure I say heterozygous, I say homozygous if you need it for that. Read everything carefully. Again, you can just copy and paste to do your Punnett square. If it's easier, you can do it on paper for this because I am more concerned about finding the correct percentages. And then copy and paste your parent genotypes in there. Star-eyed and square-eyed. I do want to point out square-eyed, this is a typo, which I need to fix. That actually should say little e, not 2 R's. There we go. Little e for eye, which means square-eyed is going to be your recessive eye shape. And I have 2 problems with that. Last one is smiley suns. It's the same concept, except now we have 2 types of rays. So we have circle rays, spiked rays, and then a bare sunny. And there are 2 examples for that as well. Again, the same thing. Put in your parent genotypes. If you want to do your Punnett square on paper and just put your percentages in, that's fine. Or you can copy and paste it into the Punnett square on the slide as well. So that is the homework assignment. That is the information with it. If you have any questions, please let me know. Have a wonderful day. Bye.